Yes, sir.
Yes. So anyone who would like to volunteer for today's summary? First, tell me, where did we start in the last class and where did we end? I think we started with transcription itself, right?
So we wrote about replication fork first.
Replication fork. I think we have done transcription also, isn't it?
Yeah, we did transcription also.
Yes, right. Okay, Arpit. How about you? Giving a summary.
Sir, I was not there in the last class. Sir, I was not ready.
Okay. Okay. Sumaira, you were there in the last class? Yeah, I think you were there.
Yes, sir.
Yes, can we go with the summary? Let's start. Yes, Sumaira, are you there?
Yes, sir.
Yeah.
So we started with
Okay.
transcription. It is a process of copying the genetic information from one strand of DNA into the RNA. Then we discussed the question why both strands of DNA they are not copied. So the first reason is that if both the strands are, if they act as a template, then they will code for two different RNA molecules with different sequence. These two different RNA molecules will code for two different proteins for the same gene, so it is not possible. So the
Yes.
two RNA molecules will be complementary to each other. And if produced together in a cell, they will form a double-stranded RNA. A double-stranded RNA cannot be translated into a protein, and the process will be futile.
Yes, right.
Then we discuss about the transcriptional unit. It has three regions: the promoter region, a structural gene, and the terminator gene. A promoter is located towards the 5' of the structural gene. It is a DNA sequence that binds the RNA polymerase. The presence of promoter in a transcriptional unit, it defines the template and the coding strand. Then the terminator, it is located towards the downstream of the structural gene, and that causes the transcriptional to end.
Yes.
Then transcription unit and the gene RNAs. There are four
And also,
types of RNA.
how is this template and coding strand decided? We discussed that as well, right?
It is from 5' to 3'.
But both the strands have 5' to 3' in some or the other direction. Remember, we were also discussing that why only one strand is the template strand and the one is the coding. So the position of promoter and terminator, that decides the direction, right?
Yes, sir.
Because transcription can only happen starting from a promoter and ending at a terminator. But still, you can say that why can't both the strands still be transcribed? So the RNA polymerase, additionally, it also has a direction of action. It can only work or synthesize new RNA in 5' to 3' direction. So automatically there are two constraints now. One, it has to start from promoter, go through the structural gene and end at terminator. Second, it has to do it in 5' to 3' direction. So automatically the strand which is in 3' to 5' from promoter to terminator becomes the template strand. Make sense?
Yes, sir.
Yeah. Okay, continue.
So there are four different types of RNA, that is mRNA, tRNA, rRNA, and snRNA.
Okay, what do we understand from these different types of RNA? After knowing that there are multiple types of RNA, what was the complexity that generated? Sorry. do all these are you there, Sumaira?
Yes, sir.
Yeah, do all these RNAs translate into proteins?
No, only the RNA gets translated into proteins.
Yes, and the rest three are functional as RNAs itself, correct?
Yes, sir.
Now the problem here is that we used to define gene as the functional unit of inheritance located on DNA, right? And we know that every gene gets transcribed into RNA. So that is clear for all the genes. But now what happens is all the genes that get transcribed into RNA may not get translated into proteins. You understand, everyone? Only
Yes, sir.
mRNA will get translated into protein. All other RNAs, tRNA, rRNA, or snRNA, they function as RNAs. Okay, they do not code for proteins. So then it became more crucial to define a gene that codes for proteins separately from the genes that do not code for proteins, which codes for RNA but do not go on to get translated into proteins. And then came the concept of cistron. Okay, Sumaira, can you continue?
Yes, sir. So a cistron, it is defined as the segment of DNA that codes for a polypeptide. There are two types of cistron, that is monosystonic structural gene and polycystronic structural gene. The
Yes.
monosystonic, it codes for only one polypeptide chain, and it is present in eukaryotes, example cats, yeast, humans, etc. And the structural gene has exons and introns also. Exons are useful and introns are waste. Then polycystronic structural gene, it codes for 3 different polypeptides, and it is found in prokaryotes.
Not 3, it can code for multiple, more than one.
Yes.
It's not decided that it will only code for 3. I gave you this example of 3. Okay, poly means, what does the word poly means?
More than one.
Multiple.
Yeah, multiple, right? And cistron means a segment of DNA that codes for a polypeptide chain. So in polycystronic gene arrangement, there is one single promoter under which multiple, so there is one structural gene. The structural gene is continuous. So if you ask me how many mRNAs are produced, and this is one question which was asked in NEET, so you can write down. Just add this point here. In a polycystronic, the polycystronic structural gene, the polycystronic structural gene transcribes into a single mRNA. It may transcribe into a single mRNA, but gives rise to multiple polypeptide chains after translation. But gives rise to multiple polypeptide chains after translation. Okay, so basically the concept is to pack more than one genes as the same structural gene unit under the same promoter. So every time if you have to, and most of the time these genes are those genes which are required together. Now try to understand this point, it is very crucial. In humans also, there are many genes. In eukaryotes, there are many genes which are required together. Is it clear? There are many genes which are required together. But for every gene in our body, we have a promoter. Okay, its own promoter. You understand, everyone?
Yes.
Yeah. So suppose we have to transcribe, let's say 3 genes or 4 genes that have to work in a pathway together. But we have to start their transcription from every promoter, their respective promoters. In case of prokaryotes, they save space by doing this. If these 3 genes, if you can see my cursor, if these 3 genes are important for a similar function or to act together in a pathway, then you better pack it as one cassette and put it under one promoter. So when RNA polymerase comes and bind to this promoter, it will transcribe all the 3 genes in one go, and you will get all the 3 polypeptides or proteins that will do the function. Is it clear? It's a more packed way, more compact way to make gene structures. Is it clear, everyone? Make sense to you? Yes, no, something.
Yes.
Yeah, okay. Okay, good. Then I ask you to go and prepare transcription in prokaryotes and eukaryotes, and also read genetic code. Did you do this? If you do this, today's class will be more beneficial to you. So let's move forward and let's talk about the first thing for today, which is transcription in prokaryotes. Okay. Transcription in prokaryotes. Now, transcription in prokaryotes is, it's simple, okay, because prokaryotes cannot afford a lot of diverse molecules in the cell. They are single unicellular organisms. All prokaryotes are unicellular, right? They also don't have a nuclear membrane, and you know all the characteristics of prokaryote. Yes or no? Yes. Good. So in
Yes.
prokaryotes also, yeah, but in prokaryotes also there are three types of, these three types of RNAs are present: mRNA, tRNA, and rRNA. Okay. So they have a mRNA, which you already know what it is. You have a tRNA, which you know what it is, and you have a rRNA. All these three RNAs are required for translation, but all these three RNAs have to be transcribed first, right? During the process of transcription. And the good thing is that in prokaryotes or in bacteria, to transcribe all these three RNA, you have a single RNA polymerase. Okay, so write down, in bacteria, there is a single DNA dependent RNA polymerase. Now you understand what these names mean, right?
Yes, sir.
Yeah, so there is a single DNA dependent RNA polymerase that transcribes all the three major types of RNAs. That transcribes all the three major types of RNAs. I've also told you the function, right, of different RNAs. Messenger, transfer, ribosomal. Okay, I've just told you the name. So let me also tell you the function now. Now you can write down mRNA. Have you written down the function already? Yes or no?
No, sir.
No,
Okay.
sir.
No, sir.
Okay. So write down mRNA provides the template for protein synthesis. Second is tRNA. It acts as transfer RNA. It acts as transfer RNA and brings amino acid. it also reads the genetic code, because both these processes are clubbed together. The molecule which can read the genetic code can only bring the amino acid. How? We will discuss in translation, but just know that tRNA brings amino acids and reads the genetic code of mRNA. Okay. And third is rRNA. Write down. It plays structural and catalytic role. Remember, I told you that when we were discussing RNA world hypothesis, I told you that RNA also has the capability to catalyze reactions.
Yes, sir.
Yes. So this rRNA, it plays the structural and catalytic role during translation, because rRNA is an integral part. It is the structural part of ribosomes. I told you that ribosomes are non-membranous organelles, right? This you have studied from your junior classes, correct?
Yes, sir.
And you also know, tell me, what do you know about ribosomes?
So they are the protein synthesis factor.
Yes, right. And about the structure, do you know anything about the structure?
They have one small part and one big part.
Yes, they have a smaller subunit and a bigger subunit, right? And in your junior classes, you have also studied the S coefficient of those subunit, which you have forgotten. So please go back and revise. So the ribosome, both the subunits, bigger and smaller, is nothing but just RNA and proteins, different type of proteins and RNA present together, bound together. Okay, and the smaller subunit binds to mRNA. So I will tell you about ribosomes in translation, but just know that rRNA is the RNA that makes the ribosome. So that's why the structural, and it also helps in this process of polypeptide synthesis, hence catalytic roles. Is it clear? Yes? No? People?
Yes, sir.
Okay, now just for a moment, I will stop the share. Do you have NCRT, everyone?
Yes.
Yes, sir.
Please open your NCRTs and come to page number. I'm telling you, just let me pull this page. 109. Page number 109. Can you
Yes,
see
sir.
the figure of transcription there?
Yes, sir.
So,
Yeah.
Yes, sir. There's 3 steps, right?
Yeah, the process of transcription in bacteria. Let me pull out that figure for you. Can you see that figure, the process of transcription in bacteria?
yes, sir.
Okay. And just 1 second. Okay. I've got the figure. Can you see the screen that I'm sharing, everyone? Yes?
Yes.
Okay, so this is the figure. Yes. Yes, is it visible to all of you? Let me make it a little bigger. Yes, can you see this?
Yes, sir.
Yes.
Very good. Okay, now I have told you that for all different kinds of RNA, there is only one RNA polymerase. Now there are 3 steps in the transcription, both of eukaryotes and prokaryotes, and it goes by the name initiation. So you have to start any process, then elongation, where the mRNA is formed or the RNA is formed, and finally termination, where it stops. Now what happens is, uh, so I told you about the promoter and the terminator region, right? So can you see the promoter region here marked in yellow, this region? This is the promoter region of the DNA, right?
Yes.
And RNA polymerase will bind to which strand? 3' to 5' or 5' to 3'?
3' to 5'.
Yes, so basically it is this strand that it will bind to. I could make it this one, right? Which I have marked in black. Can you see this?
Yes, sir.
So this region, this yellow marked region, it will bind to. Now that is the promoter region. They have shown it here. Now this is RNA polymerase, and this is a sigma factor. It is also known as initiation factor. It is a protein. Basically, the point is, how will the RNA polymerase know where to begin from, where to bind in the DNA? Now for that, this sigma factor or initiation factor comes into picture. So initiation factor, it binds to RNA polymerase momentarily, just for a moment, to guide it, just to tell it that you have to start from here. Okay. And once RNA polymerase is bound to the DNA, the sigma factor is released. And once the sigma factor is released, initiation is done, and all that RNA polymerase has to do is to keep moving and keep synthesizing RNA. So over time, is initiation clear to you? RNA polymerase, and this is all happening in bacteria. So bacteria have a simple single RNA polymerase to which a sigma factor binds. Without sigma factor binding to RNA polymerase cannot bind to the promoter region. Once it binds, then the sigma factor comes out and the RNA polymerase starts polymerizing the RNA. Okay. And it happens, transcription happens in localized bubbles. So it's not like to transcribe any region of the DNA, you will open the whole DNA. Instead, only the space which the RNA polymerase needs is opened. So as it proceeds during elongation, so you can see that from here, from the promoter region, it has proceeded this much, right? So this long mRNA has been produced. So once it transcribes and moves forward, the DNA region behind it again closes and winds back into a helical structure. And the immediate region which is in front of RNA polymerase then what you can say, opens. Is it clear?
Yes.
Yeah. So it keeps moving and keeps producing the mRNA. The moment it reaches the terminator region, can you see this region? This part here. This is the terminator region. Okay. The moment it reaches the terminator region, there is another factor that comes and binds to RNA polymerase. So basically, these factors are like switching on and switching off the RNA polymerase to bind on DNA. So if sigma binds, RNA polymerase binds to the promoter region. And the terminator factor is called Rho. So when termination factor binds, RNA polymerase falls off the DNA. And when it falls off, it also detaches from the RNA that is being produced, and the whole gene or the region from here to here is transcribed. Okay. And now this mRNA is out. ready to go into translation if it is an mRNA. If it is a tRNA or rRNA, then it will have its own functions. Is this part clear to you, everyone?
Yes, sir.
Yes. So write down. Write down. The RNA polymerase. The RNA polymerase binds to the promoter region. Binds to the promoter region by associating transiently or momentarily with initiation factor, also called as sigma factor. Okay. Transiently with initiation factor, also known as sigma factor. Is it clear? Done, everyone? Next point. It uses, so when it is elongating, when it is making more RNA, what do you need to make RNAs? What is the monomer of RNA or DNA? Nucleotides, right?
Yes, sir.
DNA or RNA is made up of small linkers called nucleotides. So next point, write down. During, okay, write down.
Sir, what was like written after it uses?
Sorry.
So what did you say after the second point, sir? It uses, sir, after the what?
Yeah, I'm again rephrasing that point to make it better. Write down. The sigma factor dissociates. Next point. The sigma factor dissociates from the RNA polymerase and elongation begins. During which the RNA polymerase uses nucleoside triphosphates. It is a slightly modified nucleotide, so it's called. You already know what is nucleoside, right? Sugar plus base. But in a nucleotide, there is only one phosphate. Yes or no? Two phosphates. Here it is nucleoside triphosphate. So this is used as a substrate. So it uses nucleoside triphosphates as substrates to synthesize the RNA. Substrate means like to use it as the raw material. It uses nucleoside triphosphates as substrates to synthesize or to polymerize the RNA. Okay. Clear, everyone? Last point, write down. When the RNA polymerase reaches the terminator region, when the RNA polymerase reaches the terminator region, it associates with the termination factor known as the Rho factor. It associates with the termination factor or the Rho factor, and the RNA polymerase falls off, and the RNA polymerase falls off the DNA. RNA polymerase falls off the DNA along with newly synthesized mRNA or RNA. Just write RNA because there can be all different types of RNAs in prokaryotes. So the RNA polymerase falls off along with the newly synthesized RNA. Is it clear? Is the whole process clear to you?
Yes, sir.
Now,
Yes, sir.
in bacteria, there is like whatever you have studied till now will come into use to develop a concept. Now tell me, what is special about bacteria, about prokaryotes? Bacteria or prokaryotes do not have a well-defined nucleus, right? Is it clear, everyone? Am I audible?
Yes.
Yeah. So bacteria do not have a well-defined nucleus. What does this mean? What does this mean? If it has no well-defined nucleus, then what? What does it mean to you, Samira? If bacteria do not have a well-defined nucleus, what does it mean to you? What do you understand by that? Come on, people, someone has to participate. It is not my revision, okay? It is for you. Arpit? Am I audible? No.
So does translation and transcription happen in like same place because they are prokaryotes?
Yes, but not because they are prokaryotes. Why do you think transcription and translation happens at the same place? What is that same place? So this is a textbook line that you know, but what does it mean to you? What does it mean that bacteria do not have a well-defined nucleus? What does it mean that a person does not have a home to live? it means that the person is homeless, right?
Yeah.
So what does it mean that a bacteria does not have a well-defined nucleus? Nucleus is the home to what?
The DNA.
Exactly. So if there is no nucleus, what happens to the DNA? It is homeless, wandering around in the
Yes.
cytoplasm, right? Yeah.
Yeah.
It is just present around a dense substance called nucleolus, but basically it is exposed in the cytoplasm. It has no walls where it can be contained and it can be protected. So it is in the cytoplasm itself, right?
Yes.
Yeah. In eukaryotes, on the other hand, DNA is very well protected inside a nuclear membrane that is double membranous. So the process of transcription, DNA dependent RNA polymerase, if it has to access the DNA, it has to go inside the nucleus, and this RNA is being formed inside the nucleus. But in a prokaryote, this RNA is formed on the street, homeless, because this DNA is just naked roaming around. So any RNA polymerase can just reach any gene, make a RNA, and that RNA will be in the cytoplasm itself, right? Because there is no boundary condition. Make sense?
Yes, sir.
So once the mRNA starts synthesizing, I'm saying start, it has not even finished. The moment the mRNA starts synthesizing, it is right away accessible to what? What are present in the cytoplasm that makes protein, that do translation?
Ribosomes.
Exactly. So the moment mRNA is forming, even before it is fully mature, it is accessible to the ribosomes, right? And the
That's true.
ribosomes are waiting. The moment mRNA starts coming out, ribosome starts translating it. So even before the mRNA synthesis has completed, protein synthesis is already on its way. Makes sense to you, everyone?
Yes, sir.
So
Yes,
this is
sir.
the meaning that transcription is coupled with translation, because there is no boundary which can define between the two. So write down. There are two reasons. Write down. In bacteria or prokaryotes, the transcription and translation are coupled.
So can you repeat again?
In bacteria or prokaryotes, I was drinking some water. In bacteria or prokaryotes, transcription and translation is coupled. Okay, due to two reasons. Due to two reasons. First is that there is no compartmentalization, or there is no separation of cytosol from the nucleus in bacteria. There is no separation of cytosol. Cytosol is also known as cytoplasm, right? In other words, you can write there is no nuclear membrane. In other words, you can write the nucleus is not well defined. In other words, you can write there is no compartmentalization in a bacterial cell. So this word often comes, no compartmentalization. No compartments are there in a bacterial cell. In a eukaryotic cell, there are many compartments. Mitochondria is one compartment, ER is another compartment, Golgi bodies is another compartment, because all these things are membranous, right? There is no membranous organelle in a prokaryote, correct? Junior classes?
Yes, sir.
So it's all just one space. Okay. So this is one reason. And the second reason is, write down, the mRNA in a bacteria, the newly synthesized mRNA, it is also known as the nascent mRNA. You can also write this term. Nascent means new. The nascent mRNA does not require any post transcriptional processing. Any post transcriptional processing to become active. This happens in eukaryotes. So both these two things are not a barrier in prokaryotes. So the mRNA which is synthesized is an active mRNA. It can right away from the moment of synthesis act as a template, okay, and can be translated. Second, there is no compartment, so everything is happening in the same compartment. So ribosomes are also present at the same place where the DNA is present. Clear, everyone?
Yes, sir.
Yeah, makes sense. So due to these two reasons, if the question come why, you can write down this question. So it can be asked in your school examination or in board. Give reasons why transcription is coupled with translation in bacteria. So these are the two reasons you will give. Okay, done. Shall we move forward?
Yes.
Okay. So from transcription in eukaryotes, now let's come to, sorry, transcription in prokaryotes. Now let's come to transcription in eukaryotes. Now in eukaryotes, there are complexities. The first complexity is, remember I told you that in prokaryotes, to transcribe all different types of RNAs, there is only one RNA polymerase. Yes? This is not the case in eukaryotes. Clear?
Yes, sir.
Yeah, so write down, in eukaryotes, there are two additional complexities. Two additional complexities. First complexity is that there are 3 RNA polymerases. There are 3 RNA polymerases in the nucleus. Now this is also an interesting thing with prokaryotes. Do you know that prokaryotes have some organelles which are membrane bound, and not only membrane bound, they also have their own DNA inside that membrane?
Yeah, sir, like
Can
mitochondria.
perfect. Mitochondria. Can you give me any other example apart from mitochondria in a eukaryotic cell?
So what was the question, sir?
In eukaryotes, what are the cell organelles that contains its own DNA inside the membrane? One is mitochondria. The other is plastids. Have you heard about chloroplast, plastids?
Yeah, sir, chloroplast.
Yes. So chloroplasts, which are a type of plastids, and mitochondria, these two organelles have their own DNA. Now the question is, if they have their own DNA, what they must be doing with that DNA? So there are three things in a eukaryotic cell. Let's take a plant cell because plastids are not present in animal cell. So I'm taking plant cell for example. Plant cell has all the three: nucleus, mitochondria, and plastids, chloroplast. All these three organelles have their own DNA inside, right? The DNA which is present in the nucleus is called the genomic DNA because it is the cell's complete genome, complete blueprint. But what is the DNA which is present in mitochondria and plastids? What is that DNA doing? The answer is very simple. What any DNA can do. What can a DNA do? What does a DNA do? Codes for
It codes
genes.
for genes. Yeah.
Exactly. So these DNA which are present in chloroplast or mitochondria also are coding for genes. Now, before I go there, let me tell you one thing. See, if you hesitate in the class that your answer might be wrong, then you are at a loss because you're not utilizing the class. The class is not to listen to or learn things. You can read. I'm not teaching you anything which is not in your book or which is not in any textbook. I'm not telling you about my novel research that I have done. So it's not your obligation to come and sit in my class to know something. And I assume that you people are not so dumb that if someone does not tells it to you, you cannot understand that information from the book. So the whole purpose of a class is that one person will lead the discussion and that person is called a teacher and everyone else will follow the discussion and those are called students. And the roles are interchangeable. At times you can also lead a discussion which I can follow. Okay. So if you're not doing that in a class, the whole purpose of a class is just a waste because before the exam, you will have to again open your NCRT and do this whole thing again. So if you participate in a class, you might save some time for yourself because you discussed it actively. Now it's a part of your memory. You don't have to go back to understand that thing. So don't make my classes degenerate. Okay. So that once whatever I teach you before the exam, you forget it and again go back to the NCRT. Okay, everyone?
Yes, sir.
So do not worry about answers. Give me all the wrong answers. Finish all your wrong answers quota in the class so that only answers you are left with in your mind are the right ones. Okay, cool. So the DNA of mitochondria and plastids also are coding for genes, correct?
Yes, sir.
So they must also be transcribing, correct?
Yes, sir. Yes, sir.
And for them to transcribe, there also must be RNA polymerases present in these organelles, right?
Yes, sir.
Of their own. Yeah.
Yes, sir.
So there are RNA polymerases present in mitochondria and plastids. That's why in eukaryotes, I am telling you about RNA polymerases in the nucleus because that is what we are going to study in detail. We cannot discuss all the RNA polymerases, but at least the three which are present in the nucleus. Okay, that's why I use the word nucleus because you
Yes.
might be thinking that apart from nucleus, can we have RNA polymerases somewhere else? Yes, we can have RNA polymerases in mitochondria and in plastids because there is a DNA which is waiting to be transcribed. Okay,
Yes, sir.
very well. Now these RNA polymerases are just simply named, numbered in Roman numericals. So the first one is called RNA Pol 1. RNA polymerase 1. Okay. And this one transcribes, write down. RNA polymerase 1 transcribes rRNA. rRNA. Okay. And you know that rRNA are of different S units, 28S, 18S, 5.8S, etc., right?
Yes.
Yeah.
Yeah.
So just to go into some details, which is not very crucial for you to remember, the rRNA that it transcribes is 18S, 28S, and 5.8S, also known as 6S. Sometimes they write it as 6S, but it is 5.8S. These three rRNAs are transcribed by RNA polymerase 1. And since I'm telling you this, also write down here, it is a neat question which is not given in NCERT, that rRNA is the most abundant RNA in an animal cell. Write down, rRNA is the most abundant RNA in an animal cell, which clearly tells you that RNA polymerase 1 is the most abundant RNA polymerase in the animal cell.
Yeah.
Not necessarily. Okay, so it's not
Why
correlated.
not, sir?
Because one RNA polymerase can transcribe multiple rRNAs from the same gene. It will bind at promoter, make RNA, fall off, again bind at promoter, make RNA, fall off, again bind at promoter, make RNA, fall off. So even a single RNA polymerase can make 10 RNAs. So just by looking at the number of RNA, we cannot say that the polymerase that codes for that RNA is the most abundant polymerase. Okay?
Okay, sir.
Simple, right? You have to make a photocopy. Same machine can make multiple photocopies. What is making RNA? It is copying the information which is present in the DNA, right?
Yes.
Okay, very good. The second RNA which is present in the nucleus of a eukaryote is RNA Pol 2. And this one codes for, it transcribes a precursor of mRNA. Precursor means a molecule which is there but not ready as an mRNA. A precursor of mRNA. So it does not codes directly for a mRNA. In fact, it is impossible to make a mRNA directly in a eukaryotic system. I will tell you why later, but just know that it transcribes a precursor of mRNA known as yes. Am I audible to you, Sahel?
Yes, sir.
Yeah. So just be with me in the class, and in the last 5 minutes I'll talk to you. Okay. I
Okay,
know
sir.
that you have missed a few classes, but I just want to first finish this thing and then I'll talk to you. Don't worry about that. Okay.
Sure, sir.
I hope you're understanding, right? This part at least,
Yeah,
what we are
yeah.
talking about. Very good.
Yes, sir, I'm trying.
Okay, very good. If you have any doubts, please ask. Okay, no hesitations.
Sure, sir.
Yeah. Okay, and keep your enthusiasm up, folks, because I am a little low today. The weather is changing and we all are getting fever, headaches, etc. Okay, where was I? Yes, RNA
So you want to know the
polymerase.
precursor?
Yes, it transcribes a precursor of mRNA known as hnRNA. Now, what is this hnRNA? You don't have to worry so many RNAs to remember. It is simply HN is called heterogeneous nuclear RNA. HNRNA is, what does the word hetero means? Whenever you are at doubt in biology, go to the term. The term itself will answer you. Hetero means
Different.
different, heterogeneous, mixture. Remember? Class 10th or 9th.
Yes.
Heterogeneous nuclear. The word nuclear says that this RNA is only present in the nucleus, right? RNA. So heterogeneous nuclear RNA is, remember what kind of gene arrangement do eukaryotes have? When it comes to the structural gene, what kind of arrangement it has with respect to cistrons? Monosystronic or polycystronic?
Monosystolic.
Monosystronic. So Mrna must be remembering because she only revised it. Monosystronic. And you know that in monosystronic there was one line written that in monosystronic the gene is split into two things, introns and exons, right? Go back up. If I go back up for you, let me go back up. Where was it? Yes, can you see here monosystronic structural gene present in eukaryotes, also known as split gene arrangement. The structural gene has exons and introns. So when it will be transcribed into RNA, it will also have exons and introns. Yes or no? Because
Yes, sir.
RNA polymerase just copies the information. If your information is split, then it will be copied as split, right? So the RNA which will be transcribed also will have exons and introns. And I told you exons are useful and introns are waste, which means to make a mature mRNA which can directly be used by the ribosomes for protein synthesis, that only contains exons. All the introns have to be removed from it. Yes, this is one complexity with the eukaryotes. You understood? That's why RNA polymerase 2, which codes for mRNA, it does not codes for mRNA directly. It first transcribes a precursor of mRNA known as hnRNA, which is called heterogeneous nuclear RNA. Now you can write down heterogeneous nuclear RNA contains both exons and introns. Heterogeneous nuclear RNA contains both exons and introns. And in order to become a mature mRNA, it needs to go through, it means hnRNA, it needs to go through post-transcriptional processing. It needs to go through post-transcriptional processing to become mRNA. Now we will study about that
Post-transcriptional
processing to become mRNA. So you see that in prokaryotes, all the three important RNAs, mRNA, tRNA, and rRNA, were transcribed by the same polymerase. Here, rRNA is transcribed by polymerase 1, mRNA in a way is transcribed by polymerase 2, and the third one, you can guess now, is called RNA polymerase 3. And what will it be transcribing? What is left? This transcribes tRNA, right? rRNA and mRNA are done, so only tRNA is left, right? So RNA pol 3 transcribes tRNA, and it also transcribes snRNA, small nuclear RNA. Again, the word says nuclear RNAs. So it's clear that they stay in the nucleus. They are small nuclear RNAs. Okay. And there is one more thing which used to be asked earlier. I have not seen this question in NEET. Remember, all the rRNAs, 18S, 28S, and 5.8S are transcribed by RNA polymerase 1. There is one ribosomal RNA which is called 5S ribosomal RNA. This 5S rRNA is transcribed by polymerase 3, just an exception. Okay. Just to remember, if you can, it's good, but just remember these three things. Most important is, even if you forget the others, don't forget this one: mRNA, rRNA, and tRNA, because these three are important for the whole central dogma. Okay, everyone?
Okay, sir.
Yes.
So this is the first complexity, that there are 3 RNA polymerases. Okay, you all are clear about all the 3? Polymerase 1, polymerase 2, and polymerase 3, and what they code for. The second complexity is, write down. The second complexity is that the primary transcripts, the word transcript is what is for used for RNA, simple. So here the primary transcript is hnRNA, remember, right?
Yes, sir.
Then when it gets mature, it's called the secondary or the mature transcript, which is mRNA. So the primary transcript contains both exons and introns. It contains both exons and introns. Okay.
Okay, sir.
And you can write down introns are non-functional. So introns are non-functional. So if you translate introns when you are making protein, the protein will be non-functional. Okay. So this mRNA, this HN, this primary transcript or this hnRNA, write down, it has to go through post-transcriptional processing or modification. Okay. And this post-transcriptional modification are of 3 types, which happens to an mRNA. First is, first problem is, see, the moment mRNA starts coming out in a eukaryote, it cannot get translated, right? So transcription and translation is clearly not coupled in eukaryotes. Make sense to you?
Yes, sir.
Because this transcription is happening in nucleus, and before it can undergo translation, it has to pass through the nucleus, come out in the cytoplasm, then go to either the rough endoplasmic reticulum, where ribosomes are present, so that they can bind to the mRNA and start reading it. So there is a time gap between the RNA synthesis and RNA translation. Yes. And one thing that we studied about, just give me one second. Hello, am I audible?
Yes, sir.
Yes,
Yeah.
sir.
So the first, what I was saying, yes. So there is a time gap between the synthesis of mRNA and then it coming into the cytoplasm, be accessible to ribosomes so that it can start getting translated. Yes or no? And one thing you know about RNA is not a very stable molecule. Yes or no?
Yes, sir,
It
it's
is,
not stable.
yes, it is like a food that you cook and it has a shelf life of half an hour only. So there is one way that prokaryotes do. You start eating the food the moment it is getting cooked. Very weird analogy, but I think it will help you understand. If you store the food, it will go bad very fast. Okay. So right after you cook it, either you eat it. If you have to store it, what do you have to do? What can you do?
Put it in the fridge.
Yes, that is one thing you can do. And basically when you are putting it in the refrigerator, or you can add preservatives, right? There are many food items where we use preservatives to increase its shelf life, like like pickles, right? Or other processed foods. Yes or no? Even they are
Yes, sir.
cooked, baked, or fried, we can use some preservatives to increase the shelf life. So anyways, whatever we do, what are we doing is we are trying to find ways to make it stable. Yes or no? Same thing happens in case of a mRNA, which is synthesized in a eukaryote. So you know that you have to store it for some time before it can reach the ribosomes and start getting translated. So the moment it starts coming out, it can start degrading also, because there is no coupled, ribosomes are not just waiting outside the DNA to start translating it. So the first thing that is done is capping. We put a cap, the moment the RNA comes out, we put a cap at the 5' end, because the 5' end comes first, right?
Yes, sir.
So we are putting a cap at the 5' end of hnRNA. It is not yet mRNA, so I'm not writing it as mRNA. Second thing is, we start doing, once the mRNA is fully out, we put a tail also. That's called tailing. Put a cap and put a tail. So tailing is done at 3' end of hnRNA. Okay. And the third thing which is done is, in between the cap and the tail, we have both exons and introns present. So the third thing that has to be done is splicing. Splicing means removal of introns. Okay. All the introns are removed. Now, when I read this information, the obvious question, what was the obvious question that comes to your mind? Ultimately, sorry, how to remove? Okay, that is the obvious question. You are more of a mechanistic kind of a scientist than always willing to go into the mechanisms, Fatima. Yes, very good. I was not so bright like Fatima thinking of how it is removed. I had a more fundamental question. Why does a eukaryotic gene has exons and introns to begin with? Anyways, they're not doing anything. And after you make a mRNA, you have to go on removing it. So you're just keeping it so that you can remove it at the right time. Yes or no? What is the benefit? Can you think of any benefit? Why to carry something in your gene, in your structural gene, which is introns, that anyways you do not need and is a problem? You have to go through processing. You just remove it. Okay, so I'm giving this you as a homework. Okay, everyone, write down this homework. Go and you will not be able to find the answer specifically in NCERT. So try to find through other sources. And this question is not important for your boards or entrances, but just for your curiosity to understand why our genomes are split into exons and introns. So write down, why does eukaryotic structural gene contains introns? Why does eukaryotic structural gene contains introns? which are anyways useless and has to be removed through splicing. Don't you think if we did not have introns, then we would have saved a lot of energy? No need of
Yes,
doing
sir.
splicing. Yeah.
Yeah,
And I
definitely.
always told you, yeah, and I always have told you, biology, whatever it does, it has a logic. A system would never like to be, wasting energy just because biology is very intelligent. So the one thing is clear, even if we don't know or we know it, splicing is not just a waste process, right? A mistake. And presence of introns in our structural gene is not just a coincidence, right? Yes, Fatima, I'll repeat the question. The question is, why do eukaryotic structural gene has introns? Okay, because anyways it has to get spliced and removed. So why it is there in the first place? Noted, Fatima. Done.
Yes, sir, thank you.
Yeah. Okay. Now, please come to page number 110 of your NCRT. 110. You can see a figure, right?
Yes, sir.
Which I'm trying to put here. Yes, this figure. Can you see this figure? Make it a little big. Yes, can everyone see it now?
Yes, sir.
So the process of transcription is same, it has initiation, it has elongation, and it has termination. So those three things are same. And because it makes sense, in every system, every process has to be initiated. So instead of a sigma factor, there are multiple proteins which bind together. So it's more complex. So that's why it's not a part of your syllabus. So transcription in eukaryotes does not happen through the sigma or the rho factor only. It's not as simple, because there are three different types of polymerases. Suppose on the DNA, if you have to transcribe a mRNA, which polymerase will come and bind there? Polymerase 2, right? If it has to transcribe a tRNA, then polymerase 1. Sorry, 3. And if it is the rRNA, then polymerase 1, right?
Yes, sir.
So all these different polymerases, if they are different because they have different activator and terminator factors, and they are not just one single like in prokaryotes. So that's a more complex thing, but overall it's same. So something binds to RNA polymerase, whichever RNA polymerase it is, which makes it bind to the promoter region of that structural gene. Yes, Arpit, please ask.
Sir, there is no sigma factor here in this case.
There is equivalent of sigma factors, but it's not the same sigma factor. It's not the prokaryotes sigma factor. Okay, you can, and I'm not even sure that you can call it sigma, because it's not just one single protein like it is in prokaryotes.
So, sir, like how does it initiate the process? Like how does it reach to the promoter area?
Yeah,
If
there are other proteins. No, sigma is not there. Sigma and rho are for prokaryotes, because one sigma, one rho, and one polymerase is there. So all the sigmas will bind to the same polymerase and it will bind to the promoter region. In this case, there are three different polymerases. So they will have at least three different initiators and three different terminators for these three different polymerases. Yes?
Yes.
Yeah.
Yes.
Does that answer your question, Arpit?
Yes, sir. Yes, sir.
Very good. So see here, what is the, we will be talking about the complexity here. So in this case, suppose in a similar manner, this was the promoter region. Okay, I'm writing it as P. So it bound here. Then from here to here, it is the structural gene. So I'm writing it as SG. And let's say this is where the terminator region is there. I'm writing it as T. Okay. So they are already showing you that the mRNA, sorry, and this thing here is, this body is your RNA polymerase. Now you don't know which RNA polymerase it is, but you can know if I say that this is a mRNA, if they use the word mRNA, you can tell me which RNA polymerase is this, right? It is 2 then,
Yes,
right?
sir, 2.
Yeah. So, and you see these small flower like dot things?
Yes, sir.
these are initiation factors. Can you see they are multiple here, or elongation factors, but these are proteins. So RNA pol binding proteins, RNA binding proteins. Sorry, RNA polymerase binding protein. Now you can already see that this long mRNA, long RNA, at this point of time, it will not be called a mRNA. It will be called a hnRNA. So they have named it mRNA here, which is slightly misleading. It's not appropriate to call it a mRNA before before the splicing is done. So look here. The moment it starts coming out, can you see this thing? At 5' end, there is a cap. Okay.
Yes, sir.
So at 5' end, there is a cap which is of, write down, the cap is methyl guanosine triphosphate. Methyl guanosine triphosphate, also known as MG-P. G in the center, M and triple P. MG-P, methyl guanosine triphosphate. So this is the cap, okay, at the 5' end. And you can also see this yellow part is the exon, and they also have the intron. We have to get rid of the intron. So once it comes out, the second thing that happens is at the 3' end, we have to put a tail. So the tail is polyadenylation. This is called tailing. It's simple. You just keep on adding adenylate residues.
So is the tail polyadenyl or is
Yes.
the okay.
The process is called polyadenylation because you are adding multiple adenyl groups.
Okay, sir.
Is it clear?
Yes, sir.
So this green part that you see here is actually around 200 to 300 adenylate residues. Okay. So poly A. So it will be like A, long chain containing lots of A's. Okay. The process is called polyadenylation. So it's also known as poly A tail. Clear, everyone?
Yes, sir.
Yes, sir.
So once the capping and the tailing is done, the mRNA is stable now. It can have a good shelf life. It can stay in the cytoplasm. But before sending it into the cytoplasm, where ribosomes can access it and start translating it, you have to do one final thing, which is called splicing. So this is splicing, where you take out, you can see all the orange regions are being taken out. So you take out all the introns and you join back all the exons. Now this is a mRNA, messenger RNA. So this in real sense is a mRNA. Before that, it was hnRNA. So mRNA only has exons. Is it clear? Everyone?
Yes, sir.
Yes.
Okay, please note this thing now and give me 1 minute. I'll get some water for myself. Okay, just note it down. And Sahel, be here. I'll talk to you. Okay.
So we should draw this diagram.
Sorry, what? Arpit?
So we should draw this diagram.
Yes, or you should note things down, at least the things that I have marked. But in NCRT, I think it's simple, so you can draw it. Just draw it in 1 minute. I'm just coming, okay?
Yes.
Okay. Hello everyone. Am I audible?
Yes, sir.
Yes, sir. Yes, sir.
Yes. So anyone has any doubts? So this is where I will end this. I'll end the class today. Okay.
Excuse me, sir.
Yes. Arpit.
So these timings are like permanent or it was like temporary for today?
No, so these timings will sustain because we're also planning your test series after some time. So from here onwards, you will have 1.5 hour classes with me, and I'll be giving you assignments, which I'm just seeing that almost every one of you is not regular with submitting assignments. So you are keeping it, you are getting it, but you are not submitting it back. So even if you are keeping it, I will suggest that if you're not submitting it to me, please solve it for yourself. And if you have solved it, then send it also, no problem. But please go through those assignments. Don't just keep it to collect the dust. Okay, so these timings will sustain from now onwards. Okay everyone.
Okay, sir.
Yes, and if no one has any doubt, then you all can leave except for Sahal. Sahal, you stay. Okay, others can leave if they have.
Okay, sir. Thank you,
Okay.
sir.
Bye bye. Bye bye, Arpit.
Thank you, sir.
Bye bye. Okay, Sahel, are you there?
Yes, sir, I'm there.
Yes. So yes. So how are you, Sahel? How did you? So
I'm
I think
fine, sir.
it was your first class, right? A first full-fledged class.
Yeah.
Yeah. So how did you like the did you like the
Yes, sir. I liked it, but
class?
I was not aware
You could
about.
not.
I'm not actually. Yeah,
Mm.
I'm not aware about this topic, so couldn't grasp much.
Okay, no problem. So
Yeah.
do one thing. Get back to the team from on the message from which you get these links to join the class. Just you can write back to them to give you links for recorded lectures of my previous classes. Okay.
Okay.
So you take the recorded lectures for chapter 6 at least first. Do it for
Okay.
chapter 6.
I've watched for chapter 5. Chapter 6 I'll watch.
Yes, and if you have any doubt, you can come to the class with your doubts. So in the first 10 minutes, I take doubts in the class if you have any doubts. Okay. And
Okay,
this
sir.
way we will keep bringing you to the same page. Okay, Sal? And if
Okay,
required,
sir.
we can, after you have watched the videos and you have made your doubts, if required, we can have an extra class with you.
Okay, so sure. And about the notes, will I get some notes or study material?
Yeah, so handouts, you get assignments on a daily basis, and you must be noting everything that I'm teaching you here on the board, right? Apart from that, you will be added. Are you added to the WhatsApp group?
No, sir, I don't. And
Okay, so
I'm not added to the WhatsApp group. I'll
okay,
give
so
you
just
my number right now on the
yeah,
chat.
you just give your number or you just message on the number from where you get the links to add you in the group. They will add you in the group. We have your number. Okay.
Okay.
And in that group you can find my number.